Okay, we're going to start now. I'm Dr. Schultz, the course director. We put this course on annually, and each year it changes because the subject matter keeps changing under us very quickly. You can see the participants. We have Dr. Sharma, Tandon, Sundararajan, and I might be giving his talk, and Natarajan, and we also may have a guest speaker too, Tony Aldavi. All of our presenters have no financial or other interest in the genes. Genetics, very popular, not only in ophthalmology, but in other fields. You could see what has been devoted to genetics in ophthalmology. It has taken over, and there's reasons for it. Even in the literature for the people, they want to know about genetics. What is your future? What am I made of? What will I become? Here's a major article in the New England Journal of Medicine, Genomics on the Eye, and it explains everything. The eye has a pivotal role in the evolution of human genomics. At least 90% of the genes in the human genome are expressed one way or the other in the eye. It has a very impressive footprint in the eye. A third of the Mendelian inheritance in man, it's in the eye. A small change in PAX6 in the genes can cause a little anatomic abnormality in the macula, a tiny alteration that could interfere with one's vision. Here's the article, an original article. And we can see all the genes that are involved in the eye, every part of it, the retina, cataracts, glaucoma. Look at the number of genes, either in combination or isolation. And that article is right here. Retinitis pigmentosa, Stargardt's, all genetic. Sometimes one gene, sometimes numerous, expressing itself at different times in one's life. So, to discuss the genes and to discuss the various parts of the eye, we have a very prominent panel. Dr. Sharma, who happens to be the General Secretary of the AIOS and in charge of the scientific committee. We have Dr. Tandon here, who'll talk about the genetics of corneal dystrophies. I'll probably be giving the talk for Dr. Sundararajan in glaucoma. And finally, we have Dr. Natarajan, who was very prominent, past president of the AIOS and deeply involved in retina genetics. And we may have Tony Aldavi, who is very deep in cornea genetics at UCLA, Jules Stein. So I'll start with the introduction to genetics. We'll review genetics and current gene therapy. You can see the methods, CRISPR, but let's talk about proposed gene therapy. COVID virus, no matter how devastating it was, could be helpful in the future for gene transfer. And we're going to talk about the elephant in the room, artificial intelligence. Anyway, let's get started. We'll talk about genetics, crash course. The difference in the genome from one person to the other, from me to you, it's only 0.1%. A difference between me and a chimpanzee in the genes, it's only 2%. And here's the breakdown. 22 atomic genes. The higher the number, the larger the chromosome, except for 21 and 22, they interchange, where 22 is a little bit bigger than 21. And of course, you have the sexual genes at the bottom. And here's the makeup of a chromosomes, the chromatids. In the center is the centromere, and they're numbered. You have the P and the Q arms. And then we could have the crossovers, and this can be inherited as you get accidental crossovers. And this can go through the gene line into your forebearers and into your family that follows. We can see the crossing over. And when it becomes meiosis, chromatids, it could pass on. Now, this is the breakdown genetically. We talk about DNA. DNA is really a pentose sugar, nothing more. Deoxy means that there's no oxygen. Ribo means that you have the oxygen. But when you talk about a nucleotide, you have the phosphate attached to it on carbon 5. But what really makes the difference in the DNA, and it's a misnomer because the DNA is the pentose, it's the base that's attached to it. And we'll talk about the purines and the pyrimidines. It's the base. That's what's changed. When you're changing someone's DNA, you're not changing the pentose, you're changing the base. More about that later. And here's the original article. Here's the original article by Crick. This is it. Molecular structure of the nucleic acids. And they designed, Watson and Crick, the helix formation, two of them intertwined, connected by the bases. This article appeared in Nature in 1953. We can see the connection in the lower right of the bases. They figured it out, and they did it by radiographic diffusion. That's how they figured this out. And for this, they won the Nobel Prize. I have the article with me for those that want to see it. So this is the model for it. And there's a pair of templates. Each one is complementary to the other. And they're connected with hydrogen bombs. Not bombs, bonds. And they break apart the helix, and that's how they duplicate themselves. And they mention about the base pairs. And here it is, tightly wound around a histone. And it's embedded in the chromosome. And it's broken into introns, which is where the activity is. The exons where the activity is, and the introns are just spaces. In transcription, the introns are eliminated, and you get, in this case, a messenger RNA. And the RNA, they're messengers. The DNA sets the tone. The RNA is the messenger. And then you get the nucleotides that I mentioned with the phosphate, and it's translated into amino acids and proteins. Here in the cytoplasm, the DNA transcription, the messenger RNA goes outside, and finally into the amino acids. Mitochondria, apparently they're descended from ancient bacteria, and its genome is similar, our genome is similar to the bacteria. It produces its RNA in bacteria and the energy. It's circular. It's circular, not helical. Here's the circle. And we can see that they have very few genes in it, but of all things, 3 of them refer to the eye. Leber's hereditary ocular neuropathy, CPEO, and Kern syndrome. And that is maternal. Here's the breakdown of the bases. Let's go into this. The two purines, adenine and guanine, and the pyrimidines, thymine and cytosine. Thymine and RNA turns into uracil. And the 5-carbon sugar again, the pentose. And we have, I mentioned, the deoxyribose and the ribose. And when you attach the base and you attach the phosphate, that's when the action starts. They could only hook up a purine to a pyrimidine, very specific. And then you get the codons to break down. 3 of them hook up, and you can get the reading of the sense, and then they duplicate into the antisense. And if there's any difference, then we can see what happens. Now, you have to read it. You forget the first letter, the owl and the cat ate one another, and if you remove one of those letters, the whole string changes. And that's what happens with genetics. The reading frame makes no sense also if you start at the first or third letter. You start at the second letter, and that's how you read it. And then you get the amino acids from that. And the amino acids have their symbols. And here we can see the codons that make up each of the amino acids. Usually it starts with methionine, and we can get the stop codons. These are the stop codons where the whole process stops. You cannot make the amino acid, you cannot make the protein. And this is what happens in genetics. So we have the open reading frame, and we can see how in this we have metformin, proline. We can see the string of codons that make up an amino acid. Now, if you insert another one, or you delete one of the codons, or the letters in the codon, you get a totally different amino acid, or you may get nothing at all, so you don't get your protein. And we can see it here. That's called a missense, if you change anything in that. And if you remove a single base, a deletion, everything stops. And from that, you get associated genetic diseases. You could read them yourself, including retinitis pigmentosa. On top of that, we have promoters and enhancers in the expressions. Now, the introns are called junk DNA, and they encode a new perspective. You want to eliminate them. Then we have epigenetics, where environment can change your genes. And that's very important, because Darwin was the one who talked about the evolution as a result of population and where you live and what you're exposed to. These are some molecules that attach to the DNA, and it regulates your activities and your formation. So even though a lot of the cells may have the same DNA, the skin could stay the same even with changes. And epigenetics, here we could see that a methyl group could change, and therefore you could change the genetics, epigenetics. Mitochondrial, that is only inherited through the woman. So in genome sequencing, you could identify the mutant gene for a specific therapy that you want. And DNA gene replacement. Remember, we're not talking about the pentose, we're talking about the base. And here we can see the vectors that can do that. Actually, viruses are used to penetrate, or adeno-associated viruses, lentoviruses, but it's really the adeno-associated viruses that are being used today, and here's the formations of them. So they act as a gene delivery tool, take out the malfunctioning gene and put in another one. And here's the method. That little ball on top is penetrating down through the cytoplasm, enters the nucleus, and boom, you have a new amino acid and protein. And we do this. The retina people love injecting the eye, and they could inject, they think that they're changing the genes. Certainly, the photoreceptors do not replicate. Maybe the pigment retinal epithelium can. So it's the future. You cannot inject under the macula, you do it away. Perhaps there's, today, stem cells might be more effective. So the gene delivery is really through a virus. Today, practically, the future is now. Using the herpes virus. It's been used now at Bascom Palmer for dystrophic epidermolysis bullosa, because that's a lack of collagen, and it could affect the eye too. And Crystal Biotech has developed a gene, and this is what's being used in it. This has been done at Bascom Palmer. A little boy had this procedure, multiple transplants. Still, the eye became cloudy, and the placement of these genes in eye drops, it cleared up, 2025. I say, let's take a look at the COVID virus. That could be a vector too, because it could penetrate tissue. It could penetrate. And this could be used in the future. Attach the gene to an alteration of the COVID virus, and boom, it goes into the nucleus, and you can change the gene. So change the DNA, change the species. And that was from J. Craig Venter. Computer binary code, change that. You change your sentences, change the code. And we can see that many of our conditions could have multiple gene changes. Proteomics, I won't go into this too much, but this is from gene changes. So we want to test the genes, genetic testing, and we want to reach a point in our knowledge. Now, scientists can regenerate retinal cells in mice, maybe in humans in the future. Here's a lot of the diseases that are in the retina genetically based. Codebreaker, we'll talk about CRISPR. CRISPR is another method in which a piece of the chromosome, a piece could be removed and replaced with the cutting Cas9. You can see what the clustered regularly interspaced short palindromic repeats. What does this mean? Clustered regularly interspaced short palindromic repeats. By the way, she won the Nobel Prize for her work. A palindromic repeat is a double-stranded sequence of DNA. And this way, the nucleotides is the same on each strand, but run in opposite directions, and they could replicate. And the Cas9 is really the enzyme, and number 9. And so what it does is it cuts out a sequence of the genes and replaces it with another gene. CRISPR is the future. It can cut out the cell's genome at a desired location and put in a brand new one. So double-stranded breaks, and here's the precursors to it. Okay, could we do this to embryos? Obviously, it's controversial. It has been tested on some embryos, social Darwinism. And now, let's talk about AI. A colleague of mine at Cornell in the bioengineering department, we discussed this, because the larger AI-based programs and models, it probably can revolutionize how we think about and see biology as well as genetics, and maybe identify genes as to, well, how we use it to produce. What's really cool about these papers is they really create inputs into DNA sequences. It could be a proper tool, but there could be problems. And in last month's Eye magazine from the Royal Academy of Ophthalmology is an article about the threats in academia, evaluating plagiarism and artificial intelligence detection in ChatGPT. And at Cornell, I discussed with my professors, and they can tell if a paper so well written was really from ChatGPT. So here's my references. And now jump aboard. We're off to the future. Thank you. All right, we have our next speaker. Yeah. Please bring up the paper for Dr. Tandem, and then we'll take on the others. Please. Just make sure they pull up yours.
Good afternoon, everyone. Thank you, Jerry, for inviting me to participate in this instruction course. So I'm going to take you through the genetics of corneal dystrophies. With that background, you already got, all of you are probably already into genetics, and it's nice to know what's going on and where we stand with respect to therapy. So why go for genetics? It's quite expensive, it may seem esoteric, and it could maybe be interesting to know in a particular patient what is the gene, but then what do we do after that? Also, there's a cost involved, which again, and patients are very, very hopeful. Like, you've done the test and they come and they want to know the report, and then they expect you're going to do something magnificent with that. So that is another thing which you have to deal with. Now, in a country like ours, we have our own special gene pool, you may say, or ethnic variation. There are a lot of uniqueness in the population in India. And in the field of ocular research, there's lots that can be done for molecular genetics as well as epidemiological studies, and then interventional strategies as well. We could take a holistic approach and also multidisciplinary policies. So there are certain global initiatives because, for example, research and funding opportunities where you put together the clinical, basic and operational departments. There is an Indo-US Vision Research program going on, and several institutes in India have taken the opportunity to join up with collaborators in the United States. There are certain strengths that we have and there are certain strengths that they have, and putting this together can be a win-win. So there's a DBT NEI, then there's an Asian Eye Genetics Consortium and the Global Eye Genetics Consortium. Nationally, we have, of course, for within ourselves also, we don't, if we don't have to collaborate internationally, we have sources of funding with the DBT, DST, ICMR and CSIR. Now about ethics. So ethics again, there are certain protocols to be followed. The information is supposed to be confidential, and largely the country has taken a stand that we would rather not have samples going from India abroad. We would rather have capacity building in our country itself to have the wherewithal to test and to interpret. There could be sometimes a conflict of interest and finances involved. So all of these need to be kept into account if you're going to have any collaborative plan. And then we have molecular genetics and functional analysis. This is where ophthalmologists may not be fully equipped to manage on their own, and you need to find an interested geneticist. What I found is many of the geneticists, they have not been that much motivated to work in ophthalmology because they have a whole host of diseases to work on. So there are few very limited geneticists in our country who are attuned or aligned towards ophthalmic research, and that is something can be a challenge, but one can always look around, and in your own institutes you may find somebody who is willing to work in this area. Now, a little bit about our experience, how we started off in the cornea services, the Dr. Rajendra Prasad Center. We do recognize that there are unique populations in India, and certain developments have taken place over the last 10 to 15 years. People who are not corneal specialists may have learned in their post-graduation that you have epithelial dystrophies, stromal dystrophies and endothelial dystrophies, and a whole lot of names were there. And it is only with the explosion of information which came with genetics that they were able to now categorize these disorders better. So ultimately they took the genetic report as the holy grail, and often there was an overlap. So what you would think clinically is one type of dystrophy, when you saw with the gene, I'll give you examples later on, there could be a overlap. So you could have the same gene with two different phenotypes, and sometimes a phenotype with different genes involved. So the International Cornea Classification of International Cornea Society Classification of Corneal Dystrophies was proposed in 2008, and then it was updated in 2014 and 15. So as I explained, traditionally it was anatomically defined depending on which disease was the predominant disease layer affected, and then it was revised because it learned that most dystrophies are not confined to one layer, and as scientists we like to be as accurate as possible, try to be as close to what is actually going on as possible. And then there are categories depending upon the information available for the identification of genes and chromosomes. So for those people who are wondering in the beginning that what's the use of knowing the genes, this is one very good example of how having the genetic information has considerably helped the cornea fraternity in being more organized in their diagnostic process. So as I said, here are a few examples. We have the Rees-Buckler corneal dystrophy. It affects more than the subepithelial layer. Then the macular corneal dystrophy technically now is known to affect both the stroma and the endothelium. And the other changes that the IC3D did is it excluded the Bowman's layer and Descemet's membrane. There were Bowman layer dystrophy and Descemet membrane dystrophy earlier. That has now been removed. It removed the table of genes separately and incorporated it within the classification. classification. The very big change has been in the classification of CHED. We used to have CHED type 1 and type 2, autosomal recessive and autosomal dominant, but this GED type 1, which was considered autosomal dominant, has now been considered that it's not a distinct entity at all. It is a variant of PPCD, posterior polymorphous corneal dystrophy, and then they included 3 variants of epithelial recurrent erosion dystrophy and removed the Grayson-Willebrand corneal dystrophy. Now, why did they remove these? Because they found that these were terms, and of course, at the time that these were described, great credit to the ophthalmologists who carefully watched their patients and took the trouble to characterize and define. But as science grows, we have learned more, and so therefore they found that it was overlapping and did not have to have a single eponymous entity. So, in terms of the categories, here I would like to say it's something like the levels of evidence. So, we have 4 categories. If the genetic locus is known, the gene is known, the features are well defined, then it comes as the highest category 1. Here we have the example of CHED2, which now is just called CHED, early Fuchs and PPCD 2 and 3. On the other hand, if the genetic locus is known, the gene specifically is not known, the features are well defined, then it comes in the category 2. So, CHED1 was coming in this category, but now it is being clubbed in terms of terminology with PPCD type 1. And so as you can see, PPCD is now divided. What earlier was a basket diagnosis of PPCD, now it is there, but it is divided into categories, and the late Fuchs as well. Then category 3 is the sporadic Fuchs, where genetic locus is not known, the gene is not known, the features are well defined. Now question is, why is it here? Because you don't know the genetic locus, you don't know the gene, only the features are well defined. It's still here because it has been found in families, and the evidence considered felt that it still has a place to be here. And then category 4 again, where the genetic locus is not known, gene is not known, the features are not well defined, and the example here is a pre-desmus membrane dystrophy. So here, this is freely available. You can check online, and this is the way things are as of today. Now, what is the journey from AIM? So we had felt that a lot of there was a need to put people, like-thinking people together, and we were considering that we would apply for an Indo-US grant, or at least for our own country define what are the areas that are worth working on. So we had cornea ophthalmologists and geneticists from all over the country as a national think tank and a brainstorming session to do a workshop on formulation of a national action plan. And this is also combined with low vision and rehabilitation services, because a lot of the retinal disorders, there is no treatment available, and it was important that we have to provide some kind of support to such patients, and it's very important to upscale and upgrade the facilities available. So in the introductory section of the genetics group, there was a meeting, there was discussion, and it was agreed that that 3 areas were felt as important to work on. One was CHED, because it was felt that it is unique to India. The other one was retinitis pigmentosa. So these are the 2 areas which was felt our priority. And there was a concept that maybe we should try, or we should start a genetic society, like the Society for Study of the Genetics of Ocular Diseases in India. However, simultaneously, the Asian Eye Genetics Consortium came up, and then the Global Eye Genetics Consortium. So we have not formulated our own separate society, but there was a lot of great brainstorming, and we decided that we will collaborate together. And as far as the work done nationally, there's a lot of work going on in cornea, glaucoma, optic nerve, retina, and retinoblastoma. Now, a little bit of our own experience, molecular pathogenesis underlying the corneal dystrophies. Now, the other question is, with so much already known all over the world, is there any benefit of doing studies in certain areas? So here is a few examples. So the features of corneal dystrophies, we know that they are inherited disorders, there's equal frequency in males and females, there's a gradual progression, and they're usually not associated with any prior systemic disease or inflammation. We wanted to see that what is the distribution of the corneal dystrophies in North Indian population, because there weren't any studies or any reports from the north. And this is the range of the corneal dystrophies, and we were able to find some interesting findings. And we combined it with the histopathology as well as the phenotype. And several unique mutations were found, and some of them were common, but they were first reported mutations, some variations, and certain novel mutations as well. And this work has been done along with the genetics department, Dr. Arundhati Sharma from the Department of Anatomy, the genetics lab. So we've also found a novel mutation for granular corneal dystrophy, then the Reese-Buckler corneal dystrophy. Now, if you look back, typically this would have been diagnosed as a granular corneal dystrophy, but the genetic information has helped us define what the disease is exactly. And this is an example of an Avellino corneal dystrophy. Now, I found with the genetic information, I was able to look at the clinical pattern again, and I found that when you have these linear deposits, I mean the opacities have a kind of radial pattern, that is more likely to be Avellino corneal dystrophy. And the purpose of the study was to see if we can do a genotypic and phenotypic variation and mapping to see if one can predict the gene based on the clinical parameters, but unfortunately it is not that simple, and you do require the genetic report to help you. And this is an example of a lattice corneal dystrophy. But if you see this kind of pattern, you would, the other thing that we noticed is there's a lot of change in the phenotype because of the nature of the population as well as the country. So you may have dryness, you may have associated trachoma, you may have associated infections that the patient may have had. So that may also change the phenotype. So it's very important to have a proper documentation of the history and the previous records of the patient. These are examples of another novel mutation that we found of MAP-dot fingerprint dystrophy, and these are the pictures of the sibling. And the patient had MAP-dot fingerprint dystrophy, and the sibling clinically had granular corneal dystrophy, and this was the mutation. And this is an example of macular corneal dystrophy. And then macular corneal dystrophy, as you know, is an autosomal recessive inheritance. And largely we found that the mutations were reported, though there were a few novel mutations. And you can see there are 5 novel mutations which have been discovered. And this is the molecular genetic analysis. This is an example of the novel mutation. So in conclusion, no coding region changes identified in 32 patients, both new coding, encoding as well as upstream regions. They were screened for TGF-beta-I and there was no mutation found in them. And we noticed there is genetic heterogeneity. So the coding regions, splice sites, upstream regional mutations were not seen in all our cases. Now coming to the endothelial dystrophy. So we looked at Fuchs dystrophy as well as CHED. And the number of specimens we had for CHED were 59 males and 41 females, and 39% did not have a familial history. So this is an example of the Fuchs dystrophy cases, and this is an example of CHED. And another interesting thing is that some of our patients, they have CHED, but they have reasonable vision. So they have navigational vision, and they avoid or they were never offered corneal transplants. So we had the opportunity of having patients as old as 40 or 50 years old. And they also had the secondary changes such as you can see here, the lipoid dystrophy and so on. So this is example of Fuchs. And then Descemet's thickening seen on ASOCT, subepithelial cystic spaces, gut age changes can also be picked up very nicely, and the specular changes and histopathological features of CHED as well as Fuchs. And the SLC411 gene was detected and a molecular genetic analysis was done. We did find, however, that not all of our patients we could identify the mutation, so we are further processing the samples to see if we have any a whole genome sequencing is planned, though we have found a novel mutation in 2 patients. So no mutation was identified in 21 patients, and the existence of genetic heterogeneity with overlapping cases was confirmed. And for Fuchs dystrophy, the COL8A2 gene was screened for. Total of 30 patients were recruited, and all the codings were amplified. So I'd like to acknowledge the Department of Biotechnology, for which I was the principal investigator for the project, along with Arundhati Sharma from the Department of Molecular Genetics, and the Cornea Services, Ocular Pathology, and the Department of Anatomy. With that, I'll end here. Thank you so much. Okay, let's switch off.
Fantastic. Now we're going to have Dr. Sharma give her talk also in this subject of cornea dystrophies. Cornea, of course, is the first thing we can look at. It's so easy. So when we have clouding of the cornea, we're more interested immediately on what affects vision. To look in the back of the eye, you need all these tools like an ophthalmoscope and all. Cornea is so easy. So Dr. Sharma is preparing her talk right now. At the end, we have Dr. Natarajan, and Dr. Natarajan, if you're here, you're welcome to come up here. Please do so. Perhaps I should then, while Dr. Sharma is preparing her talk.
No,
I'm
here.
Great.
Okay.
Didn't see you. It's
Thank
so
you.
dark.
Thank you, Jerry. Thanks a ton, and I think I must congratulate Dr. Jerry Schultz for this course which he does. He's now been coming to India close to 40 years. So thank you, Jerry. Hope, and I'm sure we'll keep doing this course again and again every year. So I'm going to be talking about genetics of corneal ectasia, and corneal ectasia would include the gambit of keratoconus, pellucid marginal degeneration, as well as keratoglobus. And the incidence is 1 case per 2000, and although it is mostly sporadic, the genetic basis is seen only in 10% of the cases. The prevalence is 6-fold in Asians than Caucasians, and much more in Indians than other Asians. And I think with the advent of the diagnostic tools that are there and available to us, this is increasingly being diagnosed more often as compared to what it used to be done earlier. So genetic basis of keratoconus, if you see, it has a sporadic distribution with 10% familial genetic inheritance. Autosomal recessive or autosomal dominant pattern of inheritance is also seen in different ethnicities. Rabinowitz reported 0.23 to 0.05% of keratoconus in prevalence of the general population, and 3.34% prevalence is reported in first degree relatives and concordant in monozygotic twins. If you look at the increased corneal curvature, it has been found to be an important inheritability trait, and as per the Beaver Dam Eye study in 2009 on 715 individuals in 185 families, they reported 95% prevalence. Now, the advent of newer technologies like the next generation sequencing, the GWAS, RNA sequencing and linkage mapping have showed identification of multiple gene associations in keratoconus, and the VSX1 gene is the one which is most commonly implicated. These mutations were reported in the pathogenesis of keratoconus from Australian, Indian, Caucasian, Iranian and Italians, but not from many others also. And having said this, VSX1 gene is also associated with other disorders like endothelial corneal dystrophy, fixed dystrophy and posterior polymorphous corneal dystrophy. Now, the ethnic variation, low frequency of changes and polygenic nature of the keratoconus are the primary reason for conflicting report of the pathogenicity of VSX1 in keratoconus. And although we look at these mutations, but it itself is not a suitable biomarker, but only a possible one. So this just shows the pathogenic VSX1 gene mutation and a novel mutation that we actually found in our cohort when we did a study on 50 keratoconic corneas. The mutations in the VSX1 homebox gene for two distinct corneal dystrophies are also shown here, that is the PPD and the keratoconus both, as shown by the other group of investigators. Now, this is basically to show the SOD1 gene, that is the differences in the SOD isoenzyme distribution in keratoconic corneas in the stromal region. And although brittle cornea syndrome is not linked directly to the keratoconus, but brittle cornea syndrome, patients of brittle cornea syndrome may have keratoconus, and this is just to show that it is a generalized connective tissue disorder characterized by thinning and fragility of the cornea, which can develop even after minor trauma and is associated with homozygous mutations in ZNF469. Now, the TGF beta pathway also plays an important role in the pathogenesis of keratoconus, at least in the Chinese population it has shown so. And in this study, which was done by Schultz et al., mutation in the region of MIR 184 was responsible for familial severe keratoconus combined with early onset anterior polar cataract by deep sequencing of a linkage region known to contain a mutation. Now, this is from the same study. And then we've also looked at the mitochondrial DNA in 20 keratoconic patients and 20 controls. And the mitochondrial complex were sequenced, and in our study 20 keratoconus and 20 controls DNA sequencing revealed 84 nucleotide variations in the keratoconic patients and 29 in the controls. So there are SNPs which are present in keratoconus which have been identified by the GWAS studies which are as shown. And then keratoconus also has comorbidities with multifactorial interactions which are not only because of the genetic association but also biomechanical, biochemical and environmental. And also many of the syndromes are associated with the keratoconus which also have a genetic basis. So gene therapy in keratoconus is, I think, the way to go in future, and cornea is an ideal candidate for the gene therapy because of immune privilege and because of the vector mediated gene transfer, both viral and non-viral. The future directions are that the recent molecular characterization of keratoconus because of it being inflammatory disorder as has been shown by the increase in the biomarkers, and so the limitations are that there is not a very good in vivo heritable keratoconic model which exhibits dissimilarities to the human keratoconic corneas, and in vitro keratoconic models hold promising avenue, but we do require human cell based 3D in vitro tissue engineered model for keratoconus that may be the future and that may closely reflect the properties of an in vivo disease tissue. So thank you very much for your kind attention.
We have a few minutes, so those that have questions about corneal dystrophies and the genetic basis, please raise your hand. We have the panelists here. Please ask the questions. Okay, so everyone knows everything about the genetics of corneal dystrophies. Dr. Natarajan.
I am Dr. Parveen. So I think we all have a common interest in genetics, and probably one question which a lot of us and everybody which I also come across is, if you see a patient of either a corneal dystrophy or a retinal dystrophy and you were to advise genetic test, which one first test would you advise, whether a gene panel, a whole exome sequencing, and how would you take the first step? I think that is the most common hurdle that people face.
So what one could do is, depending upon what one is suspecting clinically, one could ask for a gene panel, and if supposing you are not sure it's an atypical disease, you're not certain what it is, then you can ask for a whole exome sequence. It's the cost effect is there, but you can ask for a whole exome sequence. And if you really want to find the full panel of all the genes that the patient has, then it will be a whole genome sequence rather than exome. So a gene panel works out to be cost effective, and then they screen that first.
So would you also follow the same thing, or we do that in India, I know for sure, but other countries, in US or in UK, is the same pattern followed, that first they ask for a panel testing?
So in case you wanted to get a patient's genetic analysis done, what test would you ask for if you were in the US?
Well, we do have the easy ones, the chromosome analysis. Chromosome, each one of these bases has its own color, and that's what we had done before. It really depends upon the institution, what they have. Now it's much easier because we have the machinery to actually analyze the genes and the basis. So we're not just dependent upon the color of those various genes that we saw in the codons and in the basis. That was our old method. But now, when I went to Cornell, they had very large types of computers that could analyze it. Now I went back to Cornell, and they're almost using a laptop to do this. I said, where's your big computers? They said, nah, this laptop is good enough. And so many institutions are now able to, within their labs, do the genetic analysis of your patients. It doesn't take much tissue either.
Thank you. One more question, or rather opinion from you as well. If we were in India to make ourselves ready for the next set of clinical trials, 1, 2, 3, what steps, how should we all together go ahead with it?
So one step has already been taken, that the ICMR has come out with guidelines for gene therapy. So one can go through that, and that's a very important thing, because without that then one would not have been in a position to even consider. And they have already started gene therapy for some disorders in India. Then depending upon what's in the pipeline, because you don't know which therapy is going to be available. So if you have an idea about what's in the pipeline, then you can keep the list of your patients ready. So I would say there are two conditions. One is Leber's hereditary, Leber's congenital amaurosis, and retinitis pigmentosa. I forget which one. There's one particular gene, Stargardt's. Stargardt's disease is one, and Leber's congenital amaurosis are two, where two diseases where gene therapy is developed to a very high level, and so maybe you could just keep the list of those patients. Yeah.
Well, we're going to get on now, and I will give Dr. Sundarasen's talk on the molecular genetics of glaucoma. In case you have questions, you can ask the questions. I'll give the answers. Now, glaucoma is very interesting. So many people in the world have it. It's probably the second largest cause of blindness, but the major cause of permanent blindness. Well, cataracts, of course, are curable, and cataracts are number one. I'll give this talk for Dr. Sundarasen. This is his paper, because here we know the pathophysiology of glaucoma, and it could affect various areas, back of the optic nerve, the vascularization. What causes this genetically? The cupping, axon transport, and retinal cells, the retinal ganglion cells, and peripheral vision loss, all this. So what causes this? We can see, of course, the trabeculum. Primary open-angle glaucoma is our main interest. Angle closure glaucoma speaks for itself. But a lot of this is genetics. So we have a very complex set of genes in glaucoma, as I alluded to in my opening talk. It's genetically, physiologically, but also it's a neurodegenerative disorder, because we're dealing with the nerve fibers. And we have about 500 genes associated with the various types and areas where glaucoma occurs. Here we could have the endophenotypes, a region in the eye that goes haywire because of the genetics, causing maybe not increased pressure, but can cause loss of vision. We could have so-called normal pressure glaucoma, but the vision disappears genetically. Here we could see the breakdown, high tension glaucoma, the genes associated, the MIOC, the ABACA, while normal tension glaucoma, the OPTN is the main cause, the TRKs, and regular glaucoma. So parts of the eye in the pathway can be inflicted by different genes, the optic nerve, the trabecular meshwork, etc., etc. Juvenile open-angle glaucoma. Of course, we'll talk about retina in a few minutes with Dr. Natarajan, but the trabecular meshwork gene analysis, a group of genes could affect that. So we have that there's no single molecular pathway for primary open-angle glaucoma. Some suggest that some inflammation could be in there and senescence. I don't think that old people have senescence, but they do get glaucoma. In India, we can see the different types of genes that could affect the population in different areas. Here's the gene break for this part of India, the eastern part, and then we have the western part, all different. Here's the western part. So we could have the different types of variants and novel deletions. This is a major area, this IC16, it's a risk factor. And myelin is a causative factor too, with the cupping and with the area. So in conclusion, it's really an interesting, complex neurological disease. We are dealing with nerves. Please understand that. We think in terms of pressure, but when we do OCTs, we're looking at the nerve pattern. So it's clear from the recent studies that inflammation and senescence could be pivotal, but it's really genetic. So we have to have an opportunity to develop the novel and effective interventions also, and it may be possible with gene transfers in the future. Dr. Natarajan is going to now take us from the front of the eye to the back of the eye, for which, as I said, we need instruments. Well, with the cornea, we don't.
Thank you, Jerry. Can I have the slides? It's a pity they gave in a big hall and we have few people. No? Okay. I cannot see it here. I can see there. Yeah, no, in the screen it's not here. I have to look here. इधर नहीं आ रहा है. Thank you. So I am going to talk on genetics of retinal dystrophies and diabetic retinopathy. So the objective of the presentation is to learn the basics of already which has been covered, to learn the mechanism of genetic abnormalities, to see what is the role of genes in retinal dystrophies and in diabetic retinopathy. If you didn't have genetic mutation, we wouldn't have us. You need error to open the door to the adjacent. This is what Stephen Johnson, the American popular science author and media theorist. The basics is we have the DNA structure, the nucleotide, and these are the things which we have now labs to do them. And this is the DNA and the RNA and the microRNA. And nature of genetic abnormalities contributing to human disease, mutations in protein coding genes, the point mutations, frame shift mutation, trinucleotide repeat mutation, and alteration in protein coding genes other than mutations is alterations in the non-coding RNAs. And there are at the DNA level you have deletion, insertion, inversion. At protein level you have the missense mutation, nonsense mutation, frame shift deletion mutation, frame shift insert and mutation. In molecular genetics, either neutral or non-neutral, and chromosome mutations like the structural mutation, the deletion, duplication, inversion, insertions and translocations, and numerical mutations, the polyploidy and aneuploidy. And this is from the Robbins textbook of basic pathology. So retinal dystrophies are degenerative disease of the retina which have marked clinical and genetic heterogeneity, night or color blindness, tunnel vision, and subsequent progression to complete blindness. And genes encode proteins involved in retinal cellular structures, phototransduction, the visual cycle, and photoreceptor structure or gene regulation. When I did my postgraduate, the Parsons book, half the book were all the genetic diseases and many retinal conditions which we could not treat. But Arnold Sosby had written in his textbook, ophthalmology is the playground of genetics, because most of the genetic mutations have some connections with the eye. And this is the epidemiology of inherited retinal degeneration, the genetic basis of inherited retinal disease in 271 individuals in UK, published by the Ellingford and Barton et al. So the inherited retinal disorder, the genotype-phenotype correlation in Indian cohort and the importance of genetic testing and genetic counseling is published in Medical Ophthalmology in January 23 by Chitra Gopinath et al. And it's a very eye-opener because we need to improve our clinical diagnosis. That means patient's history, the clinical visual acuity, their anterior segment, posterior segment findings, then red-free, autofluorescence, fundus photo, and also the ancillary tests like the ERG, VEP, and also angiography if required. So I think it depends on OCT. So they have done 591 IRD patients of all Indian origin, and foveal hyperplasia, Bardet Biedl syndrome, best retinopathy and achromatopsia. Like that, they have 55.6% consanguineous history and 44.3% were non-consanguineous, and these are the distribution of the like including the juvenile retinoschisis. So these are the diseases which comes in the inherited retinal degeneration. As of today, lot of clinical trials are going on in achromatopsia, Best disease, cone rod dystrophy, choroideremia, macular dystrophy, RP, and RP has several genes involved and all types of genetic autosomal recessive, dominant, and also individual it can happen, and rod cone dystrophy. So to label it, we need Stargardt's disease. They even tried a pharmacological treatment for Stargardt's, but didn't go through. But people are working. The reason is, unless you do a good phenotype, followed by a genetic testing with the genotype, and then re-diagnose the patient, so that when the proper clinical trial which is successful can be applied to only selective patients. As of today, we have one approved FDA approved disease which I will be telling. There is achromatopsia. We can see the normal retina and achromatopsic retina, which is there in the achromatopsia.info. The rod monochromatism, autosomal recessive condition associated with genes CNGA3, B3 and GNAT2. And blue cone monochromatism, X-linked recessive mutation is on the X chromosome at OPN1W and OPN1WM. And this is a subretinal idovernal gene therapy targeting CNGA3, and there is a trial going on. And cone rod dystrophy, you have the ABCA4 CRX gene, GUCY2D autosomal dominant CRDs, and RPGR X-linked CRDs we have. And then inheritance pattern depends on the gene involved and can be autosomal dominant, autosomal recessive, or X-linked recessive. Genetic counseling is always advised. So it's better to have a clinician along with them a laboratory support. Either you do it yourself or outsource the lab, and have a genetic counseling which also can be outsourced. And congenital stationary night blindness, the photoreceptor, the retinal pigment epithelium, classically complete and there is the incomplete type. So ERG based, you have the Rix type and the Schubert-Bornstein type. And genetic counseling and genetic testing has to be done in such cases. So many times the one single question which the parent and the child or the adult can keep asking is, what are you going to do next? Because they are not only interested in diagnosis with phenotype and genotype, they'll ask what can be done. So Charles Kippen says, never say never again. You keep hope, but I always tell them, don't have false hope, because there are a lot of research going on for the last 3 decades, and some treatments have come and all patients cannot be benefited. And this is a publication which was there in the RO abstract in June 23, long-term functional rescue following a AAV-based gene therapy in canine model in LRIT3 congenital stationary night blindness. And this is the first drug which is approved by the FDA, the vortigene neprivac, which is called the Luxturna, and RP65 gene mutation are eligible to receive the target therapy. So this is the one treatment which is available now. And genes affect the phototransduction cascade, the retinoid cycle, photoreceptor structure, or other biological function of the photoreceptor and retinal pigment epithelium. Syndromic RP includes Leber congenital amaurosis, Usher syndrome, and Bardet-Biedl syndrome. And LCA is mostly an autosomal recessive hereditary condition caused by RP65 mutation. So this is the gene therapy, and they are also trying intravitreal. As of today, it's not working. And the subretinal is not just an injection. You have to do a vitreoretinal surgery, go into the subretinal space, and they are now trying robot to introduce the gene vector into the subretinal space. So modifications by external factors to DNA that turn genes on or off, and these modifications do not change the DNA sequence but are inheritable, and through methylation or through acetylation. And what is the relevance to clinical practice and future directions of epigenetics? Most of the genes causing RP must be identified. It must be possible to detect nearly all of the disease causing mutation with these genes. And here you have the group of disease, the Stargardt's, the RP, the cone rod dystrophy, Leber's congenital amaurosis, and so these are the genes and phenotype, which is as per the genes and the inheritance given, and it's there in the publication from the NIH. And choroideremia, there are a lot of work going. Fortunately, we don't see much in India, but we rarely see them. X-linked chorioretinal dystrophy, characterized by diffuse progressive degeneration of the RPE, photoreceptors and choriocapillaries, and choroideremia gene mutation is X-linked recessive. Genetic testing or through immunoblot analysis with the anti-REP1 antibody, prenatal testing for choroideremia. Men are predominantly affected due to the X-linked etiology, but women can be asymptomatic carriers or rarely can be affected by the dystrophy. That's why the pedigree chart is important, and there is a trial going on, gene therapy using an adenovirus vector for choroideremia. Stargardt's is my interest. We have a lot of Stargardt's in India, and ABCA4 gene through autosomal recessive, homozygous or compound heterozygous transmission, ATP binding cassette ABC superfamily. And they also tried a drug, fenretinide, which was used for neuroblastoma, and where we applied for a grant in the National Medical from UK with a professor in Queen's University, but we did not get through. So as of today, genetic thing is going on, restoring the ABCA4 gene function, which is there's a Stargardt Foundation which is working on it, and then this is how again you need a vitreoretinal surgery and inject the vector into the subretinal space. Best disease, which is a earlier a juvenile onset vitelliform macular dystrophy, and best gene therapy corrects a diffuse retina wide microdetachment modulated by light exposure. Central areolar choroidal dystrophy, we see a lot of them, and well-defined atrophy of RPE and the choriocapillaries. Autosomal dominant, CACD is most commonly caused by mutations in peripherin 2 gene, thanks to Retina Imaging Bank. And we so far covered inherited retinal degeneration, even though there is no treatment, but at least we should improve our way of working in the patient and have a counselor to counsel the patient, the family, and then encourage the teachers who are teaching the students not to delineate them from the rest of the normal population, and tell them there is some hope and let them lead a normal life as much as possible with their visually challenged. And genetics in diabetic retinopathy is very complex because there is no one gene involved. It's a microvascular disease of the retina, and the leading cause of visual disability in diabetic patients, polygenic disorder. Heritability has been estimated to be as high as 27% for any diabetic retinopathy, and 52% of proliferative diabetic retinopathy and advanced form of the disease. Is there any role of genetic factors in shaping susceptibility to diabetic retinopathy? Diabetic retinopathy susceptibility is heritable. High concordance of diabetic retinopathy severity among twins with both type 1 diabetes and type 2 diabetes. And here you see the A. Sharma et al. Who have published unraveling the role of genetics in the pathogenesis of diabetic retinopathy, and clustering of long-term complication in families with diabetes in their diabetes control and complication trial. The diabetes control called the DCCT trial. And these data provide the first available evidence that the severity of diabetic retinopathy is influenced by familial, possibly genetic factors, and confirmatory evidence that such factors influence the development. Even in practice, we see every diabetic patient will not get nephropathy, retinopathy, or even ischemic heart disease or stroke, I think, or diabetic foot, even though it's a peripheral small blood vessel disease. We don't know who will get what and which way combination you'll get, and I think there is some genetic basis we don't know. We are doing a long-term study with the Case Western University with a population of people living in the slums of Mumbai, where we find most of the patients do not have severe retinopathy, and we are working to find out what is that biomarker which is protecting them from getting severe retinopathy. And role of genes in diabetic retinopathy, where VEGF gene, aldose reductase gene, receptor for advanced glycation end products gene, genes in the renin angiotensin system. and no consistent genetic association with diabetic retinopathy have been found. And genome-wide association studies, the GWAS studies for diabetic retinopathy, published by Wrong et al. There you can see there are several publications on this, on the various things. So here you see the no diabetic retinopathy in a diabetic patient, and the main problem is hyperglycemia, induced inflammation, polyol pathway, AGE products, protein kinase C activation, and in early stage thickening of basement membrane, pericyte loss, endothelial cell damage, neurovascular dysfunction, mitochondrial dysfunction, blood retinal barrier breakdown, hyperpermeability, and advanced stages there is increased VEGF, neovascularization, neurodegeneration, vision threatening endpoint, retinal ischemia. If you see, it's a cascade which is happening in the diabetes, and we do not know which will aggravate what, and that's why we still don't know which is the genetic factor which is responsible. Is there any role of genes in all this pathogenesis? There is a publication by Arubdas and group. It's again published in the RO, novel genetic variants in extreme phenotypes of diabetic retinopathy called the DRGen study, and the disease causing variants in 4 gene, CD33, COL18A1, ZNF395, PLEKHG5, and identified coding sequence variants in a novel set of genes involved in the angiogenesis inflammatory pathway that contributes to diabetic retinopathy progression or protection. And here angiogenesis, these are the genes which control events, blood vessel development, VEGF signaling, and leukocyte adhesion, again the Sharma group. And the take home message, genetic testing, pedigree chart is key components in inherited retinal degeneration. Genetic counseling is vital in married couples who are at high risk of development disease in their progeny, and genetic testing in diabetic retinopathy and who are at high risk of development of retinopathy. And genetic testing give you idea for gene therapy, but never say never again to a patient. Same time tell the truth and convince the patient they should come for follow-ups so that they can me as a treasurer of International Council of Ophthalmology, we welcome you to World Ophthalmology Congress in Vancouver, and as a president of Asia Pacific, request all of you who are working on ophthalmic trauma to become a member. Thank you very much.
Well, thank you, Dr. Natarajan. That was a wonderful encyclopedia, in effect, of retinal genetic diseases, inherited retinal diseases. What you mentioned, though, was the choroid, and I believe that the choroid is an area in the back of the eye that can be genetically altered in adults. The photoreceptors, as I mentioned, they really don't duplicate, but areas that can duplicate for now are areas where perhaps with the current and future types of genetic alterations using the viruses, we can make a change in the people. If we see something happening and an analysis shows that there is a genetic mutation that hopefully can be altered with what we are now developing.
Yes.
So as I mentioned, the future is now. We have the tools, we have the instruments, and it's now to be able to substitute a good gene for the mutation gene. And when it comes to embryos and fertilization, that is a medical legal situation in the United States. There's been a lot of controversy in that as we do genetic analysis on the little embryos.
I was saying that was a very nice session, and we have to hand over now to the next team.
The next session.
We collected
the
the
speaker.
audience for you, so now you can continue with your session.
Yeah. So thank you all. And any probably questions you can write to any of us, and all of us are available on the email. Thank you.